Let's dive in. So what is quality control, or QC? It's a set of procedures designed to ensure the laboratory results meet defined standards of accuracy and reliability. It involves the use of control materials, performance monitoring, and error identification to detect and correct deviations in testing processes. QC is essential for maintaining the integrity of the laboratory results and ensuring that they are reliable for clinical decision making. Their purpose in a laboratory specifically is to detect, reduce, and correct errors in testing processes. This allows us to maintain confidence in test results and support clinical decision making. Oftentimes, in basically any situation in a clinical setting, you have to have QC pass first before you can move forward. Some key components of QC: control materials, which are known materials that we know what the concentration are or the identification of them are. Performance monitoring, so how we monitor the performance of these QCs. Error detection, so if there's any detection of any errors, any deviations, we need to be able to monitor and see that. And then if there's any deviations, we need to have corrective actions. Starting off with the control materials. These are known standards or reference materials used to verify the accuracy and precision of test results. Some examples are running a QC or running a control sample alongside patient samples to ensure the instrument has been calibrated properly. Performance monitoring is regular tracking of trends and variations in results to identify potential issues. An example of this is the Levey-Jennings charts to visualize control results over time. If you don't know what a Levey-Jennings chart is, it's often referred to as a QC chart. It's how labs often track their QC over time. And error detection, identifying and addressing discrepancies before they impact patient results. Example, flagging out of control range or out of range controls results for immediate investigation. And then corrective actions, implementing steps to resolve identified errors and prevent reoccurrence. Example of this is recalibrating equipment after detecting a systematic error in control results. So main steps are you have a known standard that you run alongside your specimens. You monitor the performance of that standard. You use that standard to detect any issues, and then you correct any issues with that standard, or any issues that you determined to use. You correct that. Types of quality control. Internal QC. These are conducted within the lab using control materials to monitor test performances daily. Or external QC. This is often referred to as PT. It's comparison of results with external benchmarks, often through proficiency testing. So internal QC is something that you perform in-house, and external is something that's an evaluation from outside. I know that it can be confusing. Some people call internal QC something that they've made in-house, and an external QC is something that was purchased. But in this reference, it means testing conducted inside the lab for internal, and then comparison for external. So control materials. These are with known values to verify the testing of accuracy. There's different types of controls. They can be positive controls, negative controls, blank or zero controls, as well as extraction controls. So positive controls typically are obviously known positive results, either with a known concentration if you're doing quantitative results, or qualitative is just known positive result. Oftentimes for molecular, you have positive and negative, usually it's qualitative result. So the primer set's in there, set for determining, say, maybe E. coli. And it's a situation where your clinical chemistry or toxicology, you have a quantitative result. So say maybe amphetamine at a concentration of 200 nanograms per ml, glucose at 100 milligrams per, what's ppm. Then Negative controls, so showing that you don't have any contamination or any positive results when a result should be negative. Blank or zero controls, those are sometimes used to zero out. Those are slightly different from negative controls because they're used as zero. They're used as a blank to show that there's no nothing in the background. And extraction controls, you can actually evaluate whether your process is working with an extraction control, either in terms of your ability to extract. So for molecular, it's the ability to extract DNA. So you have extraction controls to show that your extraction was proper, and that's the reason why you're getting a negative result. So you didn't detect the pathogen because the pathogen is not there, not because your extraction failed. Same with toxicology, you can have a hydrolysis QC that you're showing that when you're breaking off the glucuronides of like morphine or codeine, that you're properly knocking off those molecules, and then so your extraction is your hydrolysis is working, and you're not just the negative result because the analyte's not there. Frequency of QC testing. Determine testing intervals based on your text complexity, manufacturer recommendations, laboratory specific assessments. So test complexity, a test that might have less complexity might have it where you just do one test, one QC on the day of run. You may have to have a test QC per extraction. Um, Manufacturers often have recommendations for that. If it's a FDA approved test, if it's an LDT test, then that's kind of you guys decide that. Um, as well as your laboratory specific risk assessments. If you have known carryover, or if you have known instances of potential for contamination where you need to clean, maybe you have to use the same pipettes from one clean room to another, and they have to be cleaned in between. Then maybe you have you run QC in between those, in between cleaning pipettes. So it really depends on your laboratory settings. There can be different variations to that. Acceptable ranges and criteria. Define your performance limits. So, example, plus or minus 2 standard deviations. In this instance here, we have our parameter. We have glucose, our mean a milligram per deciliter, 100 milligram per deciliter, sorry. And then our acceptable range being 90 to 110 milligrams per deciliter. You can use quality control charts for Levey-Jennings for visual monitoring. For example, here's an example of a Levey-Jennings chart. The line in the middle is your mean, and then actually, do I have a laser pointer? No, that's okay. So the line in the middle is your mean, and then the purple line is 1 standard deviation, the yellow line is 2 standard deviations, and the green line is 3 standard deviations. Oftentimes you have to be within 2 standard deviations of your mean. So if you're outside that yellow line, then that's a failure and needs to be repeated. Things of that nature. You monitor these performance metrics to detect errors and then resolve them. So implement statistical tools, typically Westgard rules. If you don't know what Westgard rules are, I'm actually going to have a separate presentation for that later on. It's used for detecting outliers. It's well-established statistical measures to show that you're within a mean and your standard deviations make sense. Establish corrective actions and protocols for deviations. Here's an example of a Westgard rule. This is the 4S rule. What that means is your back-to-back sample, your back-to-back QC runs are outside 2 standard deviations twice in a row. So what that means is, you see here, your negative 2 SD and then positive 2 SD. So that means you have a standard deviation, four standard deviations. So that's the 4S rule. So so depending on what kind of Westgard rules you're following, what kind of implementing, there's a ton of rules for the chart for the tracking of these QCs. And you can choose to be more stringent or less stringent. There's no hard and fast rule for the implementation of Westgard rules. So documentation and record keeping. Log all QC activities, results, and corrective actions. Maintain these records for regulatory and accreditation compliance. So part of a good QA program is to check your Levey-Jennings weekly. Usually we have a checkbox on your maintenance log or your performance sheet to show that you've been monitoring that. But the technician should be looking at the QC every day, signing off on it, put it into a binder. It needs to pass on that day. Typically, oftentimes to pass, you may need to look at the Levey-Jennings charts. If you're within 2SD for both your low and your high QC, if that's what you're doing, if you're in like a blood lab, then it's fine. There's no need to look. But if you have one that's within 2 standard deviations, we have a sample without that's the high QC is outside 2 standard deviations, that might be acceptable based on your Levey-Jennings criteria. So then you refer to your Levey-Jennings charts and see if there's been a trend, if this is just a one-off. If it's a one-off, then your QC often can be passed. If there's a trend, then something needs to be performed to correct that action. And then that needs to be documented. Choosing the right control materials and defining clear performance criteria are critical components of an effective QC program. And then choosing the right sample to mimic patient samples is important to ensure that you're monitoring the performance. These clear performance criteria, once established, are a measurable benchmark for acceptable results, enabling timely detection of deviations. And these elements ensure that a QC process are robust, reliable, and are tailored to a lab's specific need, safeguarding test accuracy and compliance, as well as patient safety. So some examples of QC in action. Let's start off with a scenario. So we have a clinical laboratory performing blood glucose test, and they use control materials with a known glucose concentration to verify the accuracy of their analyzers. It's pretty typical. Then I clicked off something there. The lab runs the control samples at the start of each shift. The results are plotted on Levey-Jennings charts to monitor for deviations. One day, the control results fall outside the acceptable range, indicating a potential calibration issue. The lab recalibrates the analyzer and reruns the controls, confirming the issue is resolved before processing patient samples. So the outcome, this QC process ensures the patient results are reliable, accurate, and protecting the integrity of the clinical decisions based on those results. So by constantly running every day at the start of a shift, your blood glucose, your known concentration to verify the accuracy of your analyzer, and maintaining your Levey-Jennings charts to monitor deviations, when you have results that are outside the range, it helps you identify what the issue is. So based off of the issue being outside the acceptable range, they chose to recalibrate and rerun the controls. And then so once recalibration was performed, the controls came in, and then we're able to process the samples. So the purpose of that is the QC, when they fail for a reason. They don't just magically decide to be wrong. So when you have an issue, when you have these known concentrations, you can't get the right answer, then you have to recalibrate and get the right answer. And then that means that you know that you can test for patient samples. An important thing is at the start of the shift. So the start of the shift is when they run their QC. What that means is they run their QC before they run any patient samples for that day. So if their QC passes, then they can run patient samples. Because the QC did not pass, they needed to recalibrate and rerun controls to show that the QC passed. You cannot test patient samples with a failing QC. This is important for clinical setting. If you're from a forensic setting, oftentimes you'll run your QC with your set test. So you'll do your extraction, you have your QC, which is fine. That's good in a non-clinical setting. But a clinical setting, especially in terms of like an analyzer here with blood glucose, the LIS and the way the LIS and the EMRs are set up, the data goes straight from the instrument into the LIS and then is accessible to nurses, doctors, and then if the result is submitted out, then it's available to patients. But people can access that and see it. And the way the instruments are set up, they basically process it in real time and shoot out the results. So you can't run if you run without a QC, without QC passing, results will still get populated. They're still going to go somewhere. It's not like when you run on the LC and then you analyze the data and then you send out a report somewhere. No, these instruments, they're going to send data. So if you run something in a patient sample, it's going to send data out. So if you run without passing your QC, there's going to be data and there's going to be results that can be accessible, and they can be accidentally sent out to the patient, or the doctor can use those results by accident without them being completely confirmed. So QC is the backbone of reliable laboratory operations. This ensures that all tests are dependable and actionable. And by identifying and addressing issues promptly, QC minimizes errors and supports the delivery of high quality patient care. So by testing the QC at the beginning of the shift, without testing any patient samples, there's no impact on it. And so if there is an issue, so if maybe for some reason glucose can't pass, maybe you're out of calibration standard, maybe the QC lot you have isn't working, maybe it expired, maybe it was left out overnight and now it fails and you don't have another one in the container. You can't test patients that day. So it's important to. I love to have redundancy. It's important to have backups. Don't be cheap when it comes to QC materials, calibration materials, reagents, because it helps with troubleshooting. If you have things on hand, it's going to help you out a lot. But I digress. The key goals of QC: to ensure that tests are consistent over time, identify problems promptly to minimize patient impact, and maintain compliance with CLIA and accreditation standards. QC is the foundation of quality management. So without a QC, you can't truly have a QMS system. So earlier before we were talking about a QMS, without a QC, there's really not a monitor. There's really not an actionable monitor without a QC. You got to have that to be able to evaluate accuracy and precision of your test and reliability. Ultimately, the QC is central to protecting patient outcomes and upholding the integrity of laboratory operations. So let's talk about designing an effective QC program. So hopefully I glazed over what a QC is, why it's important, and what you can do with it. Now let's see about designing the program. Step 1 is to define your goals and objectives. To ensure test accuracy, precision, and reliability, meet regulatory requirements by either CLIA, CAP, or your other accreditation bodies, and support quality continuous improvement. So it's important to set your goals. If you have a molecular test and it's only a qualitative, not quantitative, then obviously accuracy is going to be more important. You're not going to have precision. You'll have reliability, but precision won't be a thing because it's qualitative. If you have if you're doing blood or serum on a chemical analyzer, those ranges, especially for like your ISE reagents, so your ions like chloride, sodium, potassium, those are going to be very, very a tight window of precision because they don't vary that much in the body. So you have to have a very, very, very tight window for detection. So those have to be very accurate with their results. And then toxicology, you're going to have a wide range of drugs for toxicology. So there's going to be a lot in there. But you may have glucuronides. You may have you may have to hydrolyze before you do your analysis. And so you may need to have controls to show this hydrolysis process is working properly. Step 2, select your appropriate control materials. Match your materials to your specific test system. That's kind of what I was talking about also. So it's not just, so for example, toxicology. If you're testing THC, THC gets glucuronized, morphine gets glucuronized. You need to test it as free morphine, especially if your analyzer or your mass spec is set to detect at the free morphine. You can't be testing the conjugated morphine. So you need to make sure that your control material is appropriately set up. Also, if you're testing the free, you don't want to purchase the glucuronide if you don't have a hydrolysis system set up. Then you want to matrix match as much as possible. So if you're using a nasal swab for a PCR test, then you need to use a nasal swab. If you're using a urine sample for UTI or STI, then you need to use a urine sample. Don't be using urine matrix for blood, serum, things like that. They should be the same matrix. So buy matrix matched whenever possible. And use multiple levels, high, normal, low, etc., for comprehensive coverage. Typically, you need to have at least two. If you're doing quantitative, you need to have at least a low and a high at the bare minimum. And then it doesn't hurt to have a third, kind of in the middle range. The more QC you have, the more you can drop. So it doesn't hurt to have more, and to cover a higher range of analytes of concentrations. Step 3, establish those performance criteria. So choosing, that's one of the things. If you decide to go with just 2 QC, it does make your performance criteria a little bit easier, but then you don't have any wiggle room to drop QC. QC has to pass every time. But part of the performance criteria is if you choose 3 QC, you can't be dropping the same QC over and over again. So set your acceptable ranges using manufacturer recommendations, historical data, and peer lab comparisons if available. So oftentimes it's going to be obvious what you should be using for slope ranges. Qualitative, that's simple. It's just going to be a positive or negative. So it's just what's your lowest possible detection. But for blood specifically, because blood, the concentration of the analytes you're detecting in blood, they're usually stay within a certain range. There's a few that wander, like PSA, testosterone, high sensitivity CRP, things like that are signals for distress and signals of issue, potential cancer markers, things like that. Those may vary and they could be higher or lower, but oftentimes, so like the sodium levels, potassium levels, glucose, they should all be within a reasonable range. And so you kind of have a range that you should be monitoring for. So you want to make sure that you're monitoring a realistic range for your patient pool. So the patients, there's a what they call a normal distribution, a normal range for the concentration of the analytes. You should be monitoring, make sure your QC makes sense within that range. You shouldn't be testing if let's say, so glucose tests out at 100. You shouldn't only be evaluating 200. Like I say, you shouldn't have a QC of 200 and 150 if 100 is normal. The opposite. If 100 is normal, you shouldn't have a QC of 50 and 100, because you're losing your accuracy, you're losing your reliability in the high end or in the low end, depending on. So the high end is that last example, with the low end in the previous example. So you don't want to lose ranges. You want to make sure you're maintaining an acceptable range. Make sure you're maintain your range is acceptable, but also you're monitoring a reasonable chunk for the population. And develop testing protocols. How frequency you should QC check. Is it daily? Is it shift based? Is it per batch? And procedures for running and interpreting these controls. So really it comes down to what you're testing for. There's kind of, I won't say there's hard and fast rules, but there's kind of a general industry standard. So when it comes to QC check, anything that has an extraction, typically you need to have a QC per extraction. So if you're doing a 96 well plate for molecular, then each extraction that you perform per day needs to have a QC. So if you're running, even if you're running 5 plates a day, then you need to have 5 QCs on each one of those plates. Shift based. So clinical chemistry is a good example. So when you're running blood, you run your blood at the beginning of your shift, your QC passes, and then you're good for the entire shift. The stability on those analytes, typically the calibration last for at minimum 8 hours, some 24 hours, some 7 days, some 14 days on some analyzers where they don't need to be calibrated. They don't require calibration, but if your QC fails, then you'll need to recalibrate. And then per batch, like I said, per extraction, typically that often happens. If you did one extraction but you're running one plate, or you're running multiple plates, so you have a kingfisher and you do 5 extractions at one time, you need to make sure you have at least an extraction QC, but sometimes you can just run a positive control with those plates. So it kind of goes by. There's wiggle room. There's no hard and fast rules, but there are general practices. If you're doing something, you should have a justification for why you're doing it, especially if it's on the lower side of stringency. So if you're kind of, oh, I only run QC on this day or this time, only when it's the full moon on the second day of September, then you gotta have a reason, justification for why you're doing it. If it doesn't make sense, then it doesn't make sense. They're not gonna buy it. But typically, there's kind of a, I don't want to say, industry standard for industry best practices, I guess. And implement statistical tools. It's important to monitor these. You're not just running QC in a vacuum. Your instrument is going to tell you things. It's going to slowly deviate. It happens. If you're not monitoring, if you're not paying attention to the deviations, you might not see it. And then all of a sudden something fails. And then you're like, okay, this failed. And then you don't have troubleshooting for why it failed. So utilize your control charts, your Levey-Jennings specifically. That's the best one, best way of doing a QC chart in a clinical setting. That's very popular. Basically, if you don't have a Levey-Jennings charts, inspectors gonna be like, why don't you have a Levey-Jennings chart? And apply rules for error detection. The most popular industry standard is the Westgard rules. Just real quick, the Westgard rules is typically you have 1S, 2S, 3S, 4S. So basically, if it's one standard, if it's 2 standard deviations, it's actually 2S is what it is. Then you can set it to where that's usually a warning side. If you have one result that's outside of a 2S, then usually you're like, okay, it's fine as long as only one, either your high or your low, is outside of 2S. If you have a 2S, then if both your high and your low are 2 standard deviations outside the mean, then you're toast. That's a failure. So your QC, something's wrong. Most likely, if you're both high, then your calibration is calling low. If you're both low, then your calibration is calling high. It means recalibration. But there's also other things. So there's the 4S. We saw that earlier, where you have a negative 2 standard deviations and positive 2 standard deviations. So you have basically what that tells you is your QC is out of control. If it's crazy high and crazy low, there's no consistency to your calibration. So something's wrong. Then there's trends where there's like 10 2S. So if you have no, it's just 10S, 10 1S. If you have 10 straight QCs, so if your low QC is 10 times, you're above on the positive side of standard deviation. So there's a trending, so it sits on one side. It's not varying. So basically, Westgard rules is statistical analysis for natural variation. So there is natural variation within your instrumentation. If it's random, then it's fine. If you're identifying trends, then it's a problem. So if there's trends, or if the random is too much, like the 4SD. So if you're positive or negative 2SD out, one's high and one's low, then there's no trend. You don't have a calibration curve. You just have a system that's throwing out numbers. So it's important to have those things. If you don't know what Westgard rule is, you should look that up. It's actually, there's a ton of literature on it. But yeah, if you guys are interested in that, I do have a presentation on it. We're not going to cover that specifically in this training, other than to talk about them. But moving on. Step 6: train your personnel. Ensure that all staff are proficient in QC procedures. Make sure all your staff know how to run QC, know what to do whenever there is a QC issue, know how to correct it perform corrective action, perform recalibration, who to contact, what to do if they cannot get QC to pass. Oftentimes, the technician needs to call the engineers. Maybe there's something they can do to troubleshoot. Maybe there's something wrong with lots. Maybe there's something wrong with the instrument itself. Maybe this is something that they've seen before. Always get help. Always get help. It's important that the staff know where they can go to get help. And providing ongoing education to address updates or changes. So manufacturers do change, especially if it's an FDA regulated test. So you have to do what the manufacturer says. If the manufacturer makes a change, makes an update, then you need to be prepared for it. This happens a lot in clinical chemistry. They change ranges, they change QCs. It happens in molecular often too. Maybe a kit changes, either extraction kit or primer set, especially if you're using pre-plated primer PCR plates. They can change their positions, they can change the wells, they can change the primer sets that they're identifying. So it's important to make sure that you're prepared for these changes. You understand. You're continuing your education, so that way you're always aware and up to date to if there are any changes. And finally, monitor and adjust. Regular review of QC performance data is important, and you can update protocols as needed to address trends or new requirements. So collecting the data is one thing, but you have to be doing something with that data. Basically, if you're not doing something with the data, there's no point in collecting it. Sometimes protocols need to be adjusted. Sometimes your processes need to be adjusted. Sometimes you need to work on your pipetting. Maybe there's an issue with monitoring the room temperature. Maybe the room stays warmer than it should be during certain days of the shift, and that affects the results, especially in the chemical analyzers. Those are terribly picky when it comes to temperature and humidity.